My name is Patsy Whalen, and my project is on the effect of Bruton's tyrosine kinase on glucose transport in the PLC gamma 2 pathway. And I worked with Dr. Douglas Conklin from UAlbany Cancer Research Center. So what is BTK? Bruton's tyrosine kinase, or BTK, is an enzyme that plays a critical role in intracellular signaling, regulating growth and proliferation. It transfers phosphate groups from adenosine triphosphate, or ATP, to different proteins within a cell, which changes their shape to their active forms, allowing them to do work within the cell. Several different types of cancer require BTK for survival because of its role in cellular growth and proliferation. This is the phospholipase C gamma 2 pathway. It's a signaling pathway that regulates calcium production and transcription factors. BTK is found in this pathway, acting as an important regulator which phosphorylates PLC gamma 2. PLC gamma 2 generates diglycerol and inositol triphosphate, which activates calcium ion release into the cytoplasm. Calcium ions are a signal that regulates glucose transport through the membrane. Because cancer cells rely heavily on glucose production for proliferation, I hypothesized that this pathway would be upregulated, meaning an increase in cellular response. In my experiment, I decided to use glycolytic enzymes, specifically the genes that encode them, as markers for glucose uptake. They're listed in blue in this diagram of the process of glycolysis. Glycolysis is the process of converting glucose into pyruvate, which is the first step of cellular respiration. The end product of cellular respiration is adenosine triphosphate, or ATP, which the cell uses for energy. This is especially significant for cancer cells because they require a large amount of energy for growth. If glucose uptake is increased, it can be assumed that cancer development will increase as well. The first glycolytic enzyme that I investigated was hexokinase. This enzyme activates glycolysis by phosphorylating glucose, forming glucose 6-phosphate. The encoding genes are HK1, HK2, and HK3. The second enzyme I observed was phosphohexose isomerase. It causes the isomerization of glucose 6-phosphate into fructose 6-phosphate. Isomerization is the process of transforming one molecule into an isomer, or a molecule with a different structure than the original. The gene that encodes this enzyme is GPI. Phosphofructokinase adds a phosphate group to fructose 6-phosphate, forming fructose 1,6 biphosphate. The encoding genes are PFKL, PFKM, and PFKFB1 through 4. Aldolase breaks down fructose 1,6 biphosphate to dihydroacetone phosphate and glyceraldehyde 3-phosphate. The genes that encode aldolase are aldo A, B, and C. Isomerase isomerizes dihydroacetone phosphate to glyceraldehyde 3-phosphate. The gene that encodes isomerase is TPI1. Glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate into 1,3-diphosphoglycerate. This is through oxidation and reduction reactions, or transferring a hydrogen as well as phosphorylation. The encoding gene is GADPH. Phosphoglycerate kinase phosphorylates 1,3-biphosphoglycerate and ADP to form 3-phosphoglycerate and ATP. The genes that encode this enzyme are PGK1 and PGK2. Phosphoglycerate mutase transfers a phosphate group from the C3 position to the C2 position to form 2-phosphoglycerate. The encoding genes are PGAM1, 2, 4, and 5. Enolase removes a water molecule from 2-phosphoglycerate, making phospholipyruvate. The gene that encodes enolase are ENO1 through 4. Pyruvate kinase transfers a phosphate group from phosphoenolpyruvate to ADP, forming pyruvate and ATP. The genes that encode this enzyme are PKLR and PGAM. I hypothesize that BTK and genes encoding glycolytic enzymes will be upregulated in the same samples of breast cancer cells because of BTK's involvement in the PLC gamma 2 pathway. Or in more simple terms, BTK's involvement in the PLC gamma 2 pathway will cause an increase in glucose uptake in breast cancer cells. I obtained my results using the CBio portal for cancer genomics, which is a large-scale genomic database. I observed the TCGA Firehose Legacy's breast tissue lineage, which contains 1,108 samples. I used a feature called OncoPrints to visualize alterations in the dataset. Each sample is an individual person's breast cancer tissues that were taken and tested for alterations, and these are shown vertically. The genes that I observed can be found to the left, and this horizontal section shows the expression of each gene in each sample. This key shows the different mutations that could occur within the samples. My experiment observed the placement and quantity of amplified samples, or the red marks. In addition to OncoPrints, I observed the co-expression of each gene and BTK. Co-expression shows which genes have mRNA protein levels correlated with the query genes. P and Q values were given to determine if the results were statistically significant. P values above 0.05 suggest that the observed data occurred due to chance. Similarly, if the Q values are greater than 0.05, the results were false positives. Figure 2 shows the oncoprint of BTK and 3 genes encoding hexokinase, HK1, 2, and 3. BTK was shown to have a 4% alteration and 2 samples with amplification. In contrast, the HK1, HK2, and HK3 genes had significantly more amplification, which did not occur in the same samples as the BTK sample. This suggests a lack of correlation between BTK and genes encoding hexokinase. However, figure 3 shows a significant co-occurrence of BTK and HK3 due to the p-value of 0.006 and q-value of 0.038, both being less than 0.05. Glucose 6-phosphate isomerase, or GPI, showed amplification and high levels of mRNA in some patients, as seen in figure 4. However, the amplification shown in BTK did not occur in the same patients. In addition, there were no significant co-occurrences found between BTK and GPI. Figure 5 shows the oncoprint of BTK, PFKL, PFKM, PFKP, and PFKFB1 through 4. Although there was significant amplification in PFKFB3 in many patients, they were not found in the same patients in which BTK was amplified. Additionally, the only significant co-occurrence was found between PFKP and PFKFB3, as shown in figure 6. Figure 7 displays the oncoprint of BTK, ALDO A, B, and C. ALDO A and ALDO C were shown to have the largest amount of amplification, although they were not found in the same patients in which BTK was amplified. ALDO A and BTK were found to have an insignificant co-occurrence with a p-value of 0.464 and a q-value of 0.928, as shown in figure 8. ALDO C and BTK's p and q values are both 1 exactly, suggesting an insignificant correlation as well. Figure 9 displays the oncoprint of BTK and TPI1. TPI1 has the largest amount of amplification, although it was not found in the same patients in which BTK was amplified. BTK and TPI1 were found to have co-occurrence with a p-value of 0.571 and a q-value of 0.753, as shown in figure 10. This co-occurrence was not significant. The oncoprint of BTK and GAPDH is displayed in figure 11. Amplification was found in both BTK and GAPDH in one patient. However, the co-occurrence was not significant, with a p and q value of 0.449. In figure 13, an oncoprint of BTK, PGK1, and PGK2 is displayed. Amplification of BTK and PGK1 are shown, but they do not occur in the same samples more than once. In figure 14, BTK and PGK1 have co-occurrence, but it is not significant because the p-value of 0.073 and the q-value is 0.22. Figure 15 shows the oncoprint of BTK, PGAM1, 2, 4, and 5. Although BTK occurs in the same sample as PGAM4, figure 16 shows that there was no significant co-occurrence of these genes because all p and q values exceed 0.05. Figure 17 displays an oncoprint of BTK, ENO1, 2, and 3, and 4. Amplification is shown in the same sample of BTK, ENO1, 2, and 3. Figure 18 shows BTK's significant co-occurrence with ENO3 and a p-value of 0.006 and a Q value of 0.03. Figure 19 shows the oncoprint of BTK, PKLR, and PKM. No samples were found with amplification of BTK and PKLR or PKM, and no significant co-occurrence was found. However, PKLR was found to have amplification in the majority of samples, which could suggest that the amplification of PLKR could contribute to breast cancer development. The results of this study suggest that the expression and mutation of the Bruton's tyrosine kinase gene does not exhibit a consistent correlation with the expression of glycolytic enzymes in invasive breast carcinoma, based on the data presented in cBioPortal. However, some co-occurrences were found between BTK and the hexokinase and enolase genes. HK3 and ENO3 were both shown to have a significant co-occurrence with BTK due to their low BMQ values. However, the lack of clear association between BTK and glycolytic enzymes indicates that BTK may not directly influence glucose transport or glycolysis in breast cancer. This lack of correlation may also be attributed to the fact that many signaling molecules can be present at unusually low levels within the cell and may not change in abundance considerably. Increases in the activity of signaling molecules may have a large effect, while the proteins that promote function within the cell often increase in abundance depending on how much work is required. Further research is needed to fully understand BTK's role in glucose metabolism and to uncover the interactions between signaling molecules and metabolic processes in cancer. These were the sources I used, and does anyone have any questions?